Okay, hey everybody. So this recording is, I'm making it on the fly um, because I wasn't planning on doing this from home, but we're making the best out of what we have to work with. So I'm going to be doing the video this way today for 5.2. So 5.2 is all about mitosis. So if you remember from yesterday, we talked about the entire cell cycle, which was the life cycle of the cell. And we talked about how the cell is a lot like a person in that they're born, they grow up, they mature, they reproduce, some more often than others. And then they grow even older and then they die. And the cells that they made, their daughter cells, take their place. So it's a lot like people in that way. But that's the entire life cycle of the cell. But today I want to focus on just one part of the cell's life cycle, which is um, the reproductive part of the cell's life cycle that we call mitosis. So mitosis is a lot like when a human female goes through pregnancy and labor and delivery of the baby. That's kind of like the process of mitosis. It's part of the life cycle of the person, or in this case, of the cell, but it is not, of course, the main part. It's just a short part of it. And then the rest of the time, the cell goes back to doing its daily routine. So I'm going to do a screen share with you guys. So I'm going to present my screen, and then I'm going to go over some pictures with you about mitosis. Okay, so this is the entire life cycle of the cell. Just to quickly recap, the first phase of the cell's life cycle is here in what we call G1 or gap 1. That's just from the time it's born which is here, until the time that the cell becomes mature enough to start reproducing, which is here. And this is the second stage called the S phase. S stands for synthesis, and that's where DNA replication happens. So remember, before the cell can split in half, it has to double the amount of DNA inside of it so that each half has the right amount of DNA. So each cell needs one full set of DNA, but if the cell is going to split in half, it has to have two full sets of DNA, so it has to double from one full set to two full sets. That way, when it splits in half, each half has one full set. Okay, and that's called DNA replication whenever it doubles the amount of DNA, and that happens here in the S phase. Okay, we're going to talk about that as well. And then it goes into the G2 phase, which is just preparing for mitosis. All three of these, G1, S, and G2, are all collectively known as interphase. And when the cell is not in interphase, that means that it is in mitosis, which is what we're going to focus on today. So let's get into that. So first of all, what is mitosis? As we mentioned yesterday briefly, that mitosis is the process of starting off with one cell with one full set of DNA. I'll explain what the 46 means here in just a second. But one cell with one full set of DNA splits in half into two cells that each have a full set of DNA of their own. Now, where the 46 comes in is 46 is the number of chromosomes that you have in your cell. Every cell has one full set of DNA, every cell in your body. One full set of DNA equals 46 chromosomes. So chromosomes are just pieces of DNA. That's all it is. So your DNA is not one long continuous strand. It's broken up into 46 chunks that all float around and mix in and mingle with each other inside the nucleus. Okay. So one full set of DNA is 46 chromosomes. And in mitosis, the cell is going to make a clone of itself. It's going to split right in half, and each half is going to have also its own 46 chromosomes. Now they're going to be identical, they're going to be exactly the same. It's going to be a clone, like I said, but they're both going to have 46. So how is that possible? How does it start off with 46 and then split in half, and then each half has 20? It should have 23, shouldn't it? Well, remember that's what I was saying in the previous slide, that before it splits in half, this 46 is actually going to double to 92. So double of 46 is 92. It's going to double up to 92 and then split in half, and each half is going to have 46 like it's supposed to. Okay. But this just shows you what it starts off with and what it ends with. It doesn't show you all the steps that happen in the middle, but that's what we're going to go over today. But before we do that, I just want to show you the 46 chromosomes. Okay, so this is what's called a karyotype. This picture that you're looking at is a karyotype. Okay, a karyotype is where you take all 46 chromosomes that are in one single cell and you match them up with their pair. You see, it's not actually 46 individual chromosomes. It is, but it's actually just like it's 2 copies of 23 different chromosomes. There's only 23 different chromosomes, but there's 46 total because each chromosome has 2 copies. It's kind of like a pair of shoes. You can have 23 pairs of shoes, but that means you have 46 total shoes. But each shoe has a pair or has another shoe that goes right with it that it matches, that goes in that pair. Okay, so just imagine that these are pairs of shoes. And they are arranged from 1 to 23, because there's 23 pair, 46 total. They're arranged from 1 to 23 in order from largest to smallest. Okay, and every pair is what we call a homologous pair. All right, so this is a homologous pair of chromosome number 5 because they're numbered 1 through 23. In chromosome number 5, this is a homologous pair of them. And in each homologous pair, one chromosome comes from mom and one comes from dad. So those are the 46 chromosomes. Now, remember, when I was telling you about how the DNA has to double before it can split, well, that's here in the S phase right here. Okay, so what we're about to talk about DNA replication, but remember, that's not part of mitosis. Mitosis is over here. What we're about to talk about with DNA replication, that happens before you get to mitosis. Okay, so before the cell can split, go through the process of splitting in half, it has to double the amount of DNA inside of it. That's called DNA replication. And that's what we're about to talk about right now. So in DNA replication, we start off with one DNA molecule. Now you can tell from here down, it's already been unzipped. So the first thing that happens in DNA replication is that the DNA molecule is unzipped. So just like your zipper, when you unzip your zipper, there is this enzyme called helicase. That's one of the enzymes that you need to know about right here. There's 2 enzymes, but we're going to talk about helicase first. So DNA helicase is this little purple bubble that you see here. And its job is to act like the unzipper, that little handle on your zipper that you grab whenever you unzip your jacket or your pants or whatever. That is like what helicase is. It's the unzipper. And it's basically, as you can see, it's separating the 2 DNA strands away from each other. It can do this because DNA is held together very loosely. So it's held together by these really weak bonds called hydrogen bonds that aren't even really bonds. It's more of just like 2 magnets that you put together. And yeah, they'll stick together as long as they're right up against each other. But as soon as you pull them apart, they're very easy to pull apart. And they won't stick back to each other unless you put them back together. So that's kind of how a hydrogen bond is. So helicase comes through and separates the 2 strands pretty easily. And once the 2 strands are separated, the second enzyme called DNA polymerase comes along. And there's actually 2 of them. You can see there's one right here, and there's one right here. And these two enzymes, one will go up this strand, and one will go up this strand. And what they're doing when they go up the strands is that they're reading what bases are on these strands, and then they're going to add the base that goes with that one. So if you remember in DNA, there's GCAT, GCAT. If you remember your base pairing rules, G always goes with C, A always goes with T. So DNA polymerase is going to go up and down each strand, and it's going to read what's on there and then add the complement. So if it reads a G, it's going to add a C. If it reads a C, it's going to add a G. If it reads an A, it's going to add a T, so on and so forth. And it's going to do that all the way down the DNA strand. So then by the time you get to the end, you have two complete DNA molecules, and each one contains two strands, one old strand and one new strand. So one of the strands in this new DNA molecule is the original one from this guy over here. And the other one, the green one, is the new one that just got added to it by DNA polymerase in step 2. All right. And then we need to talk about the stages of mitosis. All right. So once again, I'll go back to the cell's life cycle. We just talked about DNA replication that happens in the S phase. And then in G2, it's preparing for mitosis. And then we get into the actual act of dividing and cutting the cell in half to make two separate cells. And that's what happens here. So this is mitosis, all of these stages, PMAT, PMAT. So you're going to need to be able to recognize what stage of the cell's life, or I'm sorry, what stage of mitosis the cell is in based on what it looks like. And what you're looking for when you're trying to determine what stage of mitosis it's in is you're looking for the DNA, the nucleus, and this thing called the spindle. Here's the spindle right here. There's actually two of them. There's one and there's two. Now, so let's look for these three in each one. All right, so there's the DNA in prophase. In prophase, the DNA appears, the nucleus actually starts to disappear, and the spindle appears. So the nucleus, things show up and then things disappear. The DNA shows up. You normally can't see the DNA because it's too thin, but it will condense and tighten up and coil up. And then it becomes visible once it does that because it's thicker, you can actually see it. But while that's happening, the nucleus is actually disappearing. And that's important because the DNA needs to be able to move around freely inside of the cell in order for the cell to be able to split in half. And the spindles appear. The spindles are the things that are actually going to pull the DNA apart. And let's move on to metaphase. We'll talk more about that. So metaphase, let's look for these three, DNA, nucleus, spindle. Well, the DNA is in the middle. That's a dead giveaway. Metaphase is really easy to identify because you'll see the DNA lined up right in the middle of the cell. That's a dead giveaway that it's in metaphase. The spindle, well, first of all, the nucleus is gone. There is no nucleus anymore. It's been broken apart and broken down. It has disappeared. And the spindles, each one is reaching out and grabbing onto the chromosomes with these things that are called spindle fibers. These little blue lines are spindle fibers. And they're grabbing onto the chromosomes. And what they're going to do is they're each going to pull half of the chromosomes to one half of the cell, and the other one over here is going to pull the other half to the other half of the cell. In anaphase, that's the actual act of the DNA being pulled apart toward opposite ends of the cell. So remember, meta, middle, ana, away. In metaphase, DNA is in the middle. In ana, it's being pulled away. In telophase, we have DNA, one full set of DNA on each side of the cell. The nucleus starts to reappear, and the spindle fibers start to disappear. And then in cytokinesis, that's like the actual cutting apart of the two cells to separate them into two separate cells instead of one big cell. All right, so just remember G1, S, G2, PMATC. G1, SG2, PMATC. G1, SG2, PMATC. G1S and G2 are interphase, PMAT, and C are mitosis and cytokinesis.